Good morning, YouTube. Have you ever heard of massively parallel sequencing? It's a groundbreaking technique in the field of genetics that allows scientists to sequence millions of DNA strands all at once. Basically, it's like having a bunch of detectives working on a case at the same time, which means we can gather a lot more information in a shorter amount of time. Massively parallel sequencing, also known as next-generation sequencing, has revolutionized the study of genetics and has allowed us to make huge strides in our understanding of diseases, evolution, and more. MPS has numerous applications, including disease diagnosis, genetic screening, and drug discovery. So, let's dive in and learn more about this awesome technology. What is massively parallel sequencing? MPS is a high-throughput DNA sequencing technology that uses millions of small reaction chambers, also known as microreactors, to simultaneously sequence millions of DNA fragments. MPS can generate millions to billions of sequences in a single run, making it orders of magnitude faster and more cost-effective than previous technologies. The key to MPS is its ability to generate vast amounts of data from tiny amounts of DNA, enabling researchers to identify and compare genetic variants in a cost-effective and efficient manner. Sequencing versatility. One of the primary advantages of MPS is its versatility. MPS can sequence both DNA and RNA, making it useful for a wide range of applications. For example, MPS can be used to sequence the entire genome or just specific regions of interest. It can also be used to sequence entire populations or individual cells, which is particularly useful in cancer research. MPS Procedure. MPS has several steps, including DNA sample preparation, library preparation, sequencing, and data analysis. During DNA sample preparation, the DNA is extracted from the sample and fragmented into small pieces. These fragments are then attached to adapters, which allows them to bind to the surface of the sequencing chip. The adapters also contain unique molecular barcodes that help to identify the sample and distinguish it from other samples that may be sequenced on the same chip. Library generation and sequencing flow. Next, the fragmented DNA is amplified using PCR, polymerase chain reaction, to create a library of identical DNA fragments. These fragments are then loaded onto the sequencing chip, which is typically a glass slide or flow cell, and the sequencing process begins. During sequencing, fluorescently labeled nucleotides are added to the chip, and a camera captures the signal emitted by each nucleotide as it is incorporated into the growing DNA strand. This process is repeated millions of times, resulting in the generation of vast amounts of data. Post-sequencing analyses. After sequencing is complete, the data must be analyzed to identify genetic variants. This is done after sequencing is complete. The data must be analyzed to identify genetic variants. This is done using specialized software that compares the sequenced data to a reference. After sequencing is complete, the data must be analyzed to identify genetic variants. This is done using specialized software that compares the sequenced data to a reference genome or transcriptome. The software can identify single nucleotide polymorphisms, SNPs, insertions, deletions, and other types of genetic variations. Applications of MPS. MPS has numerous applications in medicine and research. In clinical settings, MPS can be used to diagnose rare genetic diseases, identify disease-causing mutations, and monitor disease progression. In research, MPS can be used to study complex diseases, such as cancer, and to identify potential drug targets. MPS can also be used in agriculture to improve crop yields and develop new varieties of crops that are resistant to pests and diseases. Summary. In conclusion, MPS has revolutionized the field of genomics by enabling researchers to sequence large amounts of DNA at a much faster and cheaper rate than traditional sequencing methods. Its versatility and speed have made it an indispensable tool in medicine, research, and agriculture. With the continued development of MPS technology, we can expect to see even more exciting applications in the future. Like and subscribe. Thanks for watching. If you enjoyed this video, please like and subscribe to our channel for more great content like this. And don't forget to leave a comment below to let us know what you thought of this video. Your feedback helps us improve and create better content for you in the future. Thanks again for your support.